Hi guys, today we're going to look at the control of gene expression and gene regulation. Before we get started, however, I need you to make sure that you understand completely protein synthesis because there's a lot of correlation between the two, and we're going to be referring to different stages of that process. I am going to flick backwards and forwards between some of the notes we did last lesson in the last video, particularly making reference to transcription occurring inside, so the creation or formation of mRNA. Inside of the cell nucleus, the modifications that occur, so the caps and the tails, in order for it to be read easily by the ribosomes. The fact that after it's been modified, it will then move through the cell nucleus and head to a ribosome where it's going to be read 3 letters at a time. Those 3 letters will code for an amino acid, and the amino acid is transported to the ribosome based on using tRNA. So tRNA will transport that amino acid to the ribosome. And once all of those amino acids are together, they will then fold to form a protein. That's a really quick recap of protein synthesis. Before we get started, we're going to have a quick recall of a few different things. So structural genes are coded DNA, and coded DNA is the instructions for building proteins, in other words, protein synthesis. The first step of protein synthesis is transcription, in which case the specific section of DNA is copied into mRNA. So when we say copied into mRNA, the DNA actually untwists, unwinds, and unzips in order for a RNA polymerase, to come along and create a one-side complementary chain of nucleotides or nuclear bases or nitrogen bases, in order to get a copy of what that code actually is. Once it's out of there, like we said, it's then modified and moved through the nucleus, nuclear membrane. Proteins. Proteins are a way that which genes are expressed. And some of these proteins are going to be cell components, hormones, enzymes, antibodies, and we've already talked about it, but proteins are made up of amino acids forming polypeptide chains. So now we know what gene expression is, and we're talking mainly with gene expression, the proteins that are formed. We need to note here that cells only express some of the genes that they carry. So every cell in the body of an organism carries a full set of DNA, with the exception of red blood cells or gamete cells. Red blood cells because they don't have a nucleus, and gametes because they only have half the amount of DNA, so that they can be combined with the other gamete to create a new combination of DNA. So with cells only expressing some of the genes they carry, it means that muscle cells will only transcribe genes that are related to muscle cell operation. They're not going to start building nerve proteins. They're only going to build muscle proteins and anything that's going to relate to the muscle cell operation. This is the expression of the genes. So what's actually being done? In order to make sure that these are only expressing some of the genes, gene regulation occurs. So gene regulation is the controlling of the genes which are expressed. So because of gene regulation, gene expression is then restricted. Controlling of which genes are expressed and in what quantities. So we also need to restrict the amount of proteins that are being produced by a cell as well. So these combined actually give rise to cell differentiation. So why there is skin cells, why there is nerve cells, why there is muscle cells, and so on. Cell differentiation. Gene regulation can occur at different stages. We're going to quickly break this into pre-transcription, so before the mRNA is built, post-transcription, after the mRNA is built, and then translational control, so once it's reached the ribosome and the protein is actually being put together. Pre-transcription, so we're talking about before the mRNA is actually formed. Regulatory genes associated with functional genes produce RNA or proteins. So it's not just structural genes or functional genes that are producing proteins. Regulatory genes in charge of regulating gene expression, also form their own proteins or RNA. These proteins or RNA function as transcription factors. Transcription factors are protein produced by a regulatory gene that controls gene expression. So basically, this flipped around. So a protein that is being produced by a regulatory gene that's going to control gene expression. That's what a transcription factor is. What do? They attach to the start of a functional gene in order to do one of two things. They're going to block RNA polymerase from binding with it and therefore blocking the gene. If RNA polymerase is blocked, the formation of the RNA or mRNA is not able to happen. So RNA polymerase will attach to the DNA, it will read it, and it will construct the mRNA strand. If this transcription factor, the protein that's produced, attaches to the start of the functional gene, it will actually block the RNA polymerase from connecting to it and therefore stopping the building of that mRNA strand. The other thing it can do is it can alter the rate of the gene is expressed. So how much of it is being expressed over what period of time. Just a quick note though, that this is sometimes also affected by the environmental factors. So not always is this as a result of a transcription factor. Sometimes it's due to the environment. So for example, if a cell, or if there's a massive influx of glucose that is detected, then the expression of insulin inside the pancreatic cells would increase. So it would upregulate. And the blocking of glucagon would downregulate in order to restore balance. So as a response to this situation, genes can also, or gene expression can also be upregulated or downregulated depending on what is required by the body. So that's pre-transcription. Let's have a look at post-transcription. So remember, this is after the mRNA strand has been formed. In this stage, a cell can control the amount of protein that is produced by a few different processes. So we've already got the mRNA strand, which means that a protein can be manufactured from it, unless we get up into this translational control. So the ways in which it can affect the amount of protein that's being produced is that it can modify the mRNA's longevity. Meaning, not longevity either, longevity might be a good way of putting this, meaning that it can affect the time in which it will live for before it is actually broken down by enzymes. So that's one way in which it can do it. The second way is non-coding DNA can alter the rate by blocking the trimming and the tailing. So we said here that mRNA is transcribed inside here. After it's transcribed, it has to be modified, where it's trimmed, capped, and tailed in order to be read by the ribosome. So if it blocks the trimming, capping, and tailing, it then becomes a useless piece of mRNA, by which case the enzymes will break down the mRNA and deteriorate it, bring it back to nothing. The next one is the introns may bind with the mRNA and block translation. So introns may bind. It says here that they do not need to match those. So you're used to seeing DNA or mRNA actually have pairs. So A goes with T, G goes with C, and complementary and vice versa. If the introns are going to bind with the mRNA, it actually doesn't have to line up perfectly. And what it does, so we know that it's been trimmed and modified, capped and tailed. With the introns that have been cut off, if they were to come around and bind with this mRNA, it then means we have a double strand of mRNA. And again, it can't be read by the ribosomes. So therefore, blocking translation and the collection and arrangement of the amino acids. The last one here for post-transcription is receptors at the nucleus membrane may block it from leaving. So I'll bring back my diagram. After it's been capped and tailed, it's now able to move through the nucleus and towards the ribosomes. Here at the nucleus, there's a nuclear membrane that's lined with receptors. In one way of controlling the amount of proteins that are being produced or the rate of expression of a gene This receptors can actually stop it or prevent the mRNA from leaving through the nucleus to reach and reaching the ribosomes. So that is the other way. The last one is the translational control. So we're talking about where the ribosomes are actually reading the mRNA strand. And this will depend on the availability of the required chemicals and enzymes. So the process requires a lot of different chemicals, it requires a lot of different aminos, and it requires a lot of different enzymes in order for it to construct these amino acids, these polypeptide chains and proteins. If it doesn't have, or if there is a lack of availability of one of the chemicals or the enzymes, the protein can't be manufactured because it doesn't have basically the ingredients to make it. So that's translational control. So there is one other thing that's going to affect gene expression, and this is epigenetic control. So I've come back over here, and I know I've jumped all over the page a little bit today, but bear with me. Epigenetic control of gene expression is a gene expression due to an epigenome. Epigenomes, each cell has its own type of epigenome, and it is a system of gene control outside of the DNA. What they are is a set of factors which affect which parts of DNA are activated. So this is something that is credited to for twins, identical twins, twins that have the exact same genome, yet look slightly differently. These differences are at the cause of different parts or different genes being expressed. Now, the genes that are expressed, or the factors, may occur as a result of different stimuli being introduced. The other thing is, the different stimuli can come from a variety of different scenarios or different reasons. It could be exposure to certain chemicals. It could be different hormone levels. It could be possibly diet related. So the epigenome that two different people have will vary greatly depending on what stimuli they are exposed to. Another thing to note is that the epigenome can change in a person along their lifetime. So one of these situations is puberty. Puberty is when there is a great exposure to hormones and the rates of them, which vary greatly between one person and the next person. So the epigenome can change. How is it that epigenomes are able to change what genes are actually expressed? Epigenomes produce chemical tags or factors known as methyl and acetyl groups, and these determine which genes are switched on and off. As well as this, they are believed to be inheritable. So somehow there's still research that's going on about this, but despite having the exact same DNA between two twins, they can still appear to be rather different, and definitely possible to tell the difference between one twin and the other one. That is credited to the epigenome expressing some genes over other ones in each person, and then the epigenome is then possibly inherited down or passed down through their generations. So that's what we're covering. That's all we need to cover today. Hope that was relatively clear, and good luck.